This is a production of Cornell University.
Thank you, Mura. It's a pleasure to be here. I'm very glad to share the things I've been researching and I've been learning this past few years about polyploids with you guys. And today we are going to discuss these models, how to apply these genetic models in this polyploid species. And first, I would just like to, just to justify why I'm working with polyploids. And that polyploids are basically organisms with multiple copies of a complete set of chromosomes. And of course, we are diploids, we have 2 copies. Polyploids have multiple copies. Here we have an example of banana, 3 copies, blackberry and blueberry and potato and rose. This is this is panicum, and this is they have 4 copies. We have 6 copies in chrysanthemum and sweet potato, and just God knows how many copies we have in sugarcane. Sugarcane is a mess. But the thing is, these multiple copies, they impose a lot of challenges to genomic analysis, because instead of just having 2 alleles segregating, you have multiple alleles segregating. And of course, this affects the practical breeding. And just right off the bat, I'm showing you what is our objective here in our group in NC State. We want to develop computational tools to the whole pipeline data analysis for polyploids. Since the raw sequence DNAs to the breeding situations, right? So we need to go through all these stages here. So we need to, from the raw DNA, we need markers, we need the linkage map, and I'll show you why we need the linkage map in this presentation. We will connect this linkage map with phenotypes and have the QTL. We can use this in selection and try to design better breeding strategies. And we have so far 2 programs that we are extending for our more complex polyploid populations. Basically, we have MapPoly and QTL-Poly. There are 2 programs, one for building linkage maps and the other one for mapping QTLs. But thinking in building maps in a more broad sense, we are not just ordering markers, we are actually trace back the inheritance of the haplotypes of these polyploids, and I'm going to show you this in detail today. And in the next phase of this research, we want to implement this software which we call GGS Poly, which means genetic and genomic selection poly for polyploids. So we are going to use all this structure of inheritance of haplotypes and these genetic informations to try to help breeders making long and short-term decisions in their breeding programs. And we are going to talk this in this seminar. So just an overview of polyploid species. So here we have our diploid species, right? Just 2 copies of the genome. But we can have 2 types of polyploid species, actually more than 2, but let's stay just with 2 species, 2 types. Here we have an autotetraploid, or an autopolyploid, when you have multiple sets from the same origin, right? And here's an example, potato, we have 4 sets, right, in a tetraploid. And here we have a fish slide for this potato. And we can have allotetraploids or allopolyploids, where we have multiple sets of the different origins. And here we have an example of the allohexaploid wheat. Here you can see we have 3 genomes, and they are they behave much more like a diploid genome in terms of segregation. The problem here is when you have an autopolyploid meiotic instance. So the meiosis of an autopolyploid is quite complex. And here I'm showing you the meiotic pairing of an autotetraploid. So you have 3 options here. You can have 2 of these homologs pairing 2 by 2. We call it bivalent. you can have this all four homologs pairing all at once. We call this tetravalent. And here you have three pairing by themselves, and we have one isolated chromosome. So these are trivalent and one univalent. So this case here, usually they yield sterile progenies. So we don't care too much about this in this context, but these two, they're quite common. And you're going to see how to use this. But and here are just some examples of some slides of meiotic cells in sweet potato. Here, as you can see, we have an hexavalent. So all chromosomes are combining here, and they can exchange arms in all these places that they are paired. So here we have another one. Here we have a tetravalent, and so on. And here we have several bivalents. And here is just a meiosis of a sugarcane. We can have several bivalents in a sugarcane, but this is, as you can see, is quite crowded and quite complex. And I think this is the most important thing to understand about polyploids. It's the segregation patterns that these complex genomes can have. So in a diploid, let's imagine an outcrossing species where you have 4 alleles, right? In a diploid, it's very simple. You can combine a gamete with A1, A2, or A1 or A2. We have 2 possible gametes, and here A3 and 4. And if you combine, you have 4 possible genotypes. This is very simple. And of course, here we have 3 possible genotypes if you have a biallelic marker. And if you have a biallelic marker, you can't distinguish between big A and small a. So these 2 guys here, they are the same in this situation. In tetraploids, we have 6 possible gametes. And this, I'm not considering something we call double reduction, where the same allele can be present in the gamete. So here we have, if we consider double reduction, we have classes like A1, and I'm not considering this here, okay? So we have 6 possible gametes in one parent, 6 possible gametes in the other parent, and you have 36 possible gametes, right? And here, if you assess this information using a biallelic marker, some of these gametes here, you can't distinguish them, right? Because you just have 2 alleles. And you have some combinations of these genotypes here. And in this case, if you have a biallelic marker with 2 doses, we call doses the number of the alternator, each one of the alleles here, or the reference allele. It doesn't matter. But in this case, let's call this 2 dose alleles. This will be the segregation. It will be 1 to 8 to 18 to 8 to 1, right? And this is for a tetraploid. And this is for an hexaploid, right? Hexaploid, we have 20 possible gametes for one parent, 20 possible gametes for the other parent. And when you combine, we have 400 possible gametes. The same thing we have, if you have 3 doses here, we can have a segregation like 1 to 18 to 99 to 164 to 99 to 18 to 1. So as you can see, this is a very complex scenario when you compare with the diploid scenario, right? So how to deal with this, right? And the first question is how to assess this information, right? And in diploids, we are very used to, of course, to genotype using GBS, right? This is the genotype of choice nowadays. So in a diploid, it's very easy to separate between the homozygous and the 2 classes and the heterozygous. You just have 3 classes here, right? And this is like how this data would look like in a real situation. But in a tetraploid, so if you think in that segregation 1 to 8 to 18 to 8 to 1, we have this type of scenario. And as you can see, it's much difficult to draw the limitation between these clouds here, right? And this in an hexaploid is even worse because you have classes like 1 and classes like 164. So how can you distinguish between all these classes in this scenario, right? So back in 2012, we proposed the model. Since 2012, there's a lot of software to do this. But in our case, we use a Bayesian approach to do this. And this is a real data for Sweetpotato. And what we do here, we use both the segregation in a population and also the ratio between the two variants. And we use this in a combined way in a Bayesian network. We maximize this Bayesian network. And then what we can do is to classify each one of these individuals here, right? And here you can see you have the two parents with this triangle and this square in the middle. Probably they are 3 doses, right? Of course, the model takes into account this. And then you have the segregation. You can classify each one of these individuals. But much more than that, you can attribute some probability for these individuals. So usually individuals that fall between clouds, you are not so certain about the genotypes of the individuals. So what you can have are probability distributions for each one of these individuals, right? And we're just expressing these probabilities in this three-dimensional graphic here. And this is just one SNP. This is real data. But we have this for all SNPs in our population. And I'm going to show the population in a second, right? But this is important to know how we assess the genetic variation in such a complex species. Nowadays, what we are trying to do, actually working with Moira, we are trying to, instead of just using these dosages, we are trying to use the haplotypes combining multiple alleles. We can do this in it's not that easy, but we can combine genome information and genetic information. And we are trying to do this right now, but we still need to do some research on that. And now, using these genotypes or these dosage genotypes, I'm going to talk about how to phase and how to map these multiple alleles, these multiple dosages in the context of a complex polyploid or any ploid level at all. Of course, we have computational limitations, but the model works for any ploid level. And there you go. The first thing we need to model when you're imagining a transmission of alleles is how the gametes are formed. Here, I'm doing a simplification. I'm choosing not to assess the double reduction, as I said to you, which is a gamete that contains the same homolog in the gamete, right? And here we are just considering bivalent parents or bivalent parents. So we can have this first bivalent parent, right? Pairing. Let's say 1 paired with 6, 2, 5, and 3, 4. Okay, this is one possibility. Another possibility would be 1, 6, 2, 4, 3, 5, and so on. We can have several possibilities, right? In this case, we can have 15 possibilities, right? But for any ploid level, you can have this number of possibilities here, right? Oh, by the way, all of this derivation of these formulas are in this paper here. Okay, I will share this. This will be in my GitHub, so you can see. Yeah, so you have this formula here, right? So for a tetraploid, this is 3. For an hexaploid, this is 15, and for an octaploid, this is 70. So as you can see, this is not linear, right? This is the problem with polyploids. If you add one more homolog, the complexity is not linear. It's not even exponential. Is another function, but it is an explosive function. And how can you deal with this complexity? So as I said to you, we have a bivalent pairing, and then imagine we have these 15 possible bivalent pairings, and then we can model each one of these pairings using their recombination, just like using the mindset of a back cross, right? So we will have the recombinant gametes and the non-recombinant gametes. We can model using this matrix here. This is a very popular matrix in genetics. And what we can do is combine each one of these matrices using something we call Kronecker product. And then it's very interesting because you can do this for any ploid level, and you have a very simple formula for any ploid level, given one possible configuration, right? And one possible, and one recombination fraction here. But in this possible configuration, we call psi, right? So here we have several psi's, right? And then given one of these psi's, we can have this formula holds, right? And L here is the number of recombinant bivalents we have between these two loci here. But of course, this is for one possible configuration. And what we can do, instead of just using one possible configuration, since I know all the possible configurations for any ploid level, what I can do is to sum over all possible configurations and given the probability of each one of these configurations. And here, since I'm assuming that I have an auto polyploid, right? All these configurations here, you have the same probability. And what I can do is to sum over all possible, this is for an hexaploid, 15 configurations for one gamete. And if I sum across all these configurations, I will have this formula here. This is for one gamete, okay? And then I can fill this whole matrix here. And as you can see, there's a pattern in this matrix. This is very important. And you have this function. And then you can have a probability of having two genotypes unconditional to the configuration you're assuming. If you have something that we call segmental polyploid, which means that some pairing are more likely than other pairing situations, you can work on this using this probability here. But this is another conversation. Anyway, so if you have this probability, what you can do is actually using standard likelihood functions to assess each one of the configurations of phasing are more likely. So here I have two situations, right? I have big A, and two small a's, big B and three small b's in one possible configuration. They are in association here, right? They're in the same homologue, these two. In this case, we have also the same thing. What you observe in your data is the same thing. You observe 2 and 1. And here you also observe 2 and 1. The question is, what is the configuration between these two dosages? And here we have the second configuration. And if you have that probability, you can assemble, you can build a likelihood function, right? You can maximize this likelihood function, obtain the recombination fraction. You can just compare what's the most likely configuration. So this will be a very easy way to do a phasing with combining multiple markers, right? And of course, if you have this, you can build a map or you can try to infer the inheritance just using this situation here. But because you have your recombination between all possible markers in your population, right? And then you can order, you can group them in chromosomes, and you can have your recombination fraction, and then you have a map. But as you saw in a previous slide, there's a lot of noise in this situation, especially in hexaploids. It's very difficult to distinguish between the noise and the signal. And then using just the information of two markers is not enough to have a good map or to have a good inheritance study. So So what's the next step? So instead of just using 2 markers, we are going to use the whole genome or the whole linkage group to infer the haplotype, the phasing of these dosages, right? So this is what you see in your data set. This is an example of an hexaploid, right? It's just a piece of chromosome, small piece, 7 markers, right? And I will have 2 doses, 1 dose, 3 doses, and so on. This is the information. What I want to achieve is this information. I have the 2 doses and the 2 first homologs, the second dose in the first homolog, and so on. I want to know how these doses are distributed. And if I know how they are distributed in this 6 homologs, I can have a real map. So here I have, this is a real map. I think this is chromosome number 1, the beginning of chromosome number 1 of Sweetpotato. And then you can have your nucleotides here, and you can see how these nucleotides are distributed in these homologs. And here we have the distance, the genetic distance. This is in centimorgans, right? In your map. So the map in a polyploid is not just this stripe with several markers, but is also the haplotypic configuration of the homologs. And the way we did this using something we called hidden Markov models, right? So a Markovian property is something that is very, very important in genetics, and it's using all across genetics. It's such an important model. And it's a very simple concept, but it's very powerful because it deals with something we call conditional independence, which means that if I know something that happens in the previous time, it's enough for my model, because the something that happens in the previous time is also conditioning to the something that happens the previous time. So this makes a chain. Of course, that here in the chromosomes, you can ask, oh, but this is not true, because the chromosomes, they have the three-dimensional structure, and you can have interactions. That's true. But as genetics shows, this is a very, very good way to model a chromosome. And it's very powerful. There are several papers showing that this is very powerful. And then I choose this option to model the polyploid genome. And I convert that unconditional probability you saw in that picture, in that slide with several matrices, into a conditional probability. So what is the probability of observing a certain genotype, right, in a specific time or is in a specific position in the genome, given the previous position? And this is in function of the recombination fraction, of course, the ploidy level, and the number of recombinant events we observed between these two positions. And what you can do here is actually combine this in a way we call backward and forward algorithm. It's I don't have time to explain here. If you want to understand this, there's these 2 publications here. This publication here is awesome. It's from a group in Birmingham in England, and this is a publication from our group. And what we do with the hidden Markov model, and why is hidden? This is the thing. The Markovian property tells you that you have a conditional independence, but why it's our model is hidden? And the short answer is because we can't access the true genotype. We just assess the dosages, and the dosages are not the true genotypes. The true genotypes are the homologs, right? The true genotypes are in the tetraploid are the 36 states. And I'm going to show you here the 36 states. So what we observe in a specific individual, imagine we have a cross between 2 parents. And these are the configurations of these dosages. 1 dose in one parent, 1 dose in the other parent, 2 doses in the other parent, 2 doses in the other parent, and so on, okay? And this is the true configuration. And for a given individual, I have in a population, in a biparental cross, like Okay, so this is a trio. Have two parents and one offspring. For a specific individual, I observe one dose, one dose, one dose, 3 doses, 1 dose, and so on. There's a way that I don't have time to explain here, but to map these dosages given a specific phasing configuration here in the parents. Okay, so for the true configuration, in the parent, this red dots here, they are the states that we should visit, meaning that I know this is 1, right? What are the states that could generate this 1? And the states are these red states here, right? So this, given this possible this configuration here. Same thing for this case here. We have 2 doses in both parents, but I observed 1 dose for that marker in the offspring, in this specific individual. What are the states that I need to visit? Are these states here. And the nice thing about these hidden Markov models, if you something we call Bauman-Welsh algorithm, which is a special case of expectation maximization algorithm, we can maximize this likelihood function. And what's going to happen is that we are going to trace the most likely path in this very complex trellis here. And this is what it's going to do. This is real, okay? So in the 0 interaction here, I have all possible states. When I go through my EM, okay, for these 15 markers, for this specific individual, imagine that this is something that the algorithm is doing for all individuals in my population. I can build the haplotype using the combination of these multiple markers, okay? So here, for this individual, the data said that I have just 1 dose. But when I use the hidden Markov model, this is not 1 dose anymore. This is AD from one parent and GH for the other parent. It's much, much more informative. And then, but how this is possible? Because I'm combining information of multiple markers using this hidden Markov model. And as I said, all the explanation, all the derivations, all the formulas, the EM are in that paper. And if you can discuss, because sometimes there's some trick points in that paper. It took me a long time just to figure out how to represent this mathematically. And we can discuss, just send me an email, we can discuss this. But the most important thing here is that we can see we have these two gametes here, ADGH, and then in a point here, it changes, right? Why it changes? Because there's a crossing over. And then I know exactly what was the crossing over. It was between G and F, because they are different here, you can see, right? And using this type of algorithm, we apply this in a experimental population. This is a beautiful population with a beautiful data set, right? And kudos to Body Olokulu. I've never worked with such a great data set. And also we have 2 very, very nice reference genomes, diploid genomes. We have these 2 parents, Beauregard and Tanzania. Beauregard is a orange flesh sweet potato, a high beta carotene, very sweet, low resistance to nematodes and something in another things. And here we have Tanzania. Tanzania is from, of course, Tanzania, and then low beta carotene, but very resistant. And we have a biparental population. And this is just a beautiful picture. And I said to Mark Soros this morning that I'm going to show this picture. This is a biparental cross. Can you imagine? They're all full sibs. They have the same parents, and they're so different. This is the beauty of the hexaploid, you know. And if you see, I don't have the picture of the leaves, but you have the variations between this heart form and this another form in the population as well. So you really can see the genetics working with your eyes here. It's such a beautiful population. And what we did here is apply our 2-point algorithm here, and then we could reconstruct all the linkage groups. And as you can see here, this is very consistent. This is beautiful linkage groups, right? In a linkage group, and this is recombination fraction, right? So in recombination fraction, you expect that you have closer markers in an ordered recombination fraction. You have markers that are close in the diagonal here, and markers that are far away in the corner of the matrix. And this is exactly what you observe here. And this is about 30,000 markers, right? And then using this in the HMM algorithm, we could build this map. So this is the map in this publication here. And I'm going to zoom in one of these linkage groups, and we have 15 linkage groups here. And I'm going to zoom in the first linkage group so I can show you the components of this map. Here we have the map, as we know. So this is what we will be representing if you just have a diploid map. It's just this. And of course, you can anchor this map in reference genomes. Here we have the trifida diploid genome, and here we have the triloba genome. As you can see, we have some rearrangements here, and we discussed this in the paper, but most of the genome is very, very consistent, as you can see here. And here we have the configuration of both parents, Beauregard and Tanzania, right? And here we have something we call genotypic information content, which is how much you can distinguish between haplotypes here. As you can see, we have the yellow here a little bit lower because these two first parts here between E and F, they are very similar, right? But here they are different, so you can distinguish between or among all these haplotypes. So we have the haplotypes of our parents, beautiful. But the most important thing is to reconstruct the haplotypes in the offspring. Why? Because we want to associate these haplotypes with phenotypes, right? And of course, in the diploids, you can, if you have such a dense genome and you have such beautiful reconstructed maps, and so you can trust this, but in your data set, right? And even you need to do some filtering, of course, but in a polyploid, the filtering is much heavier. And if you want to use your data set in the maximum, what you need to do is actually to use our hidden Markov model, or not the hidden Markov model, or some way to combine the information in the whole population. And you can reconstruct the haplotypes of each one of individuals in your offspring. So here, what is this graphic here? Here we have all the possible 8 haplotypes in one individual in a tetraploid potato, right? This is real data set, okay? If you look in this individual, this is public. You can redo the analysis, you find exactly the same thing here. If you don't use the hidden Markov model, what you see is something like this. And these are the possible 8 homologs inherited by this individual from 2 parents. I know the homologs in the parents, A, B, C, and D, E, F, G, and H. And here for this first part, this individual inherited A, inherited B with some noise here, okay? Inherited, for this part, inherited C, and for this part, inherited D. And the same for the other parent here, right? And these points here will be the recombination points. But you have a lot of noise, not a lot, but you have noise here. And when you use the hidden Markov model, what it does is exactly this. It cleans the data set because it uses the information of all the markers. And what you can do is to use a prior probability. So if you have markers that are less likely to be called correctly, you can inform this to hidden Markov model using that probability distribution I discussed some slides ago. Okay, so if you inform that, or even if you inform a global error, the hidden Markov model adjusts the history of the whole chromosome. Okay, you can say, oh, this is just a little bit of improvement, but in Sweetpotato, look, this is a very, very noisy individual. And this is also, again, a real individual in our Sweetpotato map. When you use that type of strategy, what you do is this. You still have some inconsistencies, but it's much better than this, right? And of course, if you use this, you deflate the map. Because if you use something like this to construct your map, it will count a lot of recombinations that are not true, right? And then you use the hidden Markov model, you deflate your map because you're counting just the true recombination fractions, okay? So this is the power of this multi-locus analysis. And again, this is developed for any ploid level. And of course, sometimes it's very beautiful to have all this graphics and statistics, but you need a software to apply this. And we have this software. It's an R package to construct these maps. It's in its third version now. And we are very glad that we have a lot of downloads, this package, and we are constantly trying to improve this package. And if you guys use and you find bugs, or if you want something new in the package, some new features, just send me an email. Actually, send an email to Gabriel, and then he will do that. Okay, which is the postdoc is working here with us. But anyway, this is MapPoly, and we can do all this analysis in MapPoly. And this is the output of one of the maps of our maps. We have a Shiny app where you can navigate in your map, and today is much more developed. And I will show you another program that we also developed to do this. But you can choose the windows of your map, and you can check possible haplotypes. And yeah, anyway, this is the map. But in polyploids, it's very interesting if you could understand the meiotic behavior or the meiotic landscape of your species. What do I mean about that? Is you need to know the points of recombinations and how the haplotypes were combined or paired in your meiosis. And using this strategy, you can do this. So here, we have the same graphic that I was showing you in some slides ago. So this is one individual in our biparental population, and I'm just studying the meiosis in this one individual. Actually, I'm studying the meiosis in the parents of the individual, in the gametes they were formed to form this individual, right? Here we have the Central American region, which we assess using the diploid genome. Hopefully in 4 or 6 months, we will have the hexaploid genome. We are working on that. And here you have the points of recombination, right? So here I have homolog A, recombination with homolog B, homolog E, recombination with C, and the whole chromosome and homolog F. So I can say that this was the configuration. I have my centromere here, and the recombination points were here. In the other parent, in the Tanzania parent, I had my homolog K, a recombination with H, and then H recombined with L, and L recombined with I, and so on. So this is very weird because we have multiple recombinations, and we actually form a chain of recombinations. And if you have a chain of recombinations, the only way to get this chain is through something we call a multivalent formation. Do you remember in the slides that I said we have a bivalent formation and a multivalent formation? And this is an evidence of a multivalent formation. And in this case, we have an hexavalent formation, meaning that each arm of the chromosomes combine to other arm of the chromosomes in exchanged pieces. You see here, the only arms that do not exchange pieces are these two arms. And then here, as you can see, if you don't have a, oops, yeah, this is good. Yeah. So here you have the result of this meiosis. And here, as you can see, for the first three homologs here, you just have two colors, right? Because we just have bivalence. But in this case, we can have three colors. Why? Because we have multivalence. So the multivalent formations is one more source of variability in your individual, right? So, and this is very important to know. And we assess all the individuals, and this is the distribution in sweet potato. These are probably bivalents, right? The light greens and lighter greens, right? And these other ones here, they are usually probably tetravalents, hexavalents, pentavalents, and so on. But as you can see, it's mostly bivalent, right? Anyway, so this is one very important thing we have here. And this is not using cytological techniques. This is very important because this fish techniques or Gish techniques, they're very labor intense to do, you know. And here is not as exact, it's not as the same thing, but it's like a shadow of that we can obtain in a cytogenetic studies. Another very, very important thing is to assess if the homologs, if there's some preferentiality in the pairing of these homologs. And we also can assess this using our probabilities, the probability of our homologs. And here, the dashed line is what you expect in a random pairing. And here we have all possible combinations of our homologs. These are our bivalents. And here, as you can see, we just have one possible configuration that is more likely here, but actually we have the reciprocal, and we notice that this is something just in this data set. It's something, probably this is something spurious here, but what we can say that in terms of segregation, sweet potato is vastly auto-polyploid. Not in terms of origin, we are not on that because this is just how the homologs are combining. The terms of origin, we need another studies, but in terms of recombination, in terms of pairing, is the same thing. So what we do here is just to have this haplotypes reconstructed, and why we do this is to connect with phenotypes, right? I don't have the time, and this is not we are starting to work more deeper in the genotype, in these phenotypes now here, but still we have several papers with our collaborators about phenotypes. As I said, this will be public. You can look into these papers, but we used all these technologies I showed you here to publish in these papers. So these are sweet potato papers, and these are potato papers with our collaborators from Africa and here in the United States as well. And this is the most important paper, which proposed the method to do this. It's a very nice paper. So we published in 2020. And now the next step we are doing here is just not using two populations, but using multiple interconnected populations. This is the first, I see here that, yeah, almost 15 minutes. Yeah, I think I'm good. And here what we are doing is just, instead of using two populations, we are using multiple interconnected populations, meaning that some parents are shared between populations, and you can reconstruct the genotypes and tell, and you can have the mosaic of haplotypes for each individual in this multiple population thing, right? Like a diallele, you have, actually you are working in an 8 by 8 diallele now, 8 by 8 diallele with hexaploids. So can you imagine how many alleles we can have in this scenario? So when we are working on that. So as you can see here, we have the pieces. Green pieces are from this species here, with New Kawogo, is another African cultivar. And then we can trace back in the same way we did in the biparental, right? And this is the map. And just instead of having just two homologs, we have multiple homology groups here instead. Here we have just three, because these three populations, we have this just three parents, but in further studies, we have much more. And I think I'm going to my slide. Oh yeah, it's not a last slide, but it's one of the last slides. And of course, we are trying to implement friendly tools, because as you can imagine, these analyses are very, very time consuming. You need a lot of computational power to do this, but we are trying to have this user-friendly tools. We have just submitted a paper for this tool here. It's a graphical tool. It doesn't do any analysis, but you can put all your results here. You can navigate your chromosomes. You can navigate your QTL peaks. We are going to put another folder here with genomic selection and probably GWAS. And then you can connect with your genome. This is something that is just a graphical, nice thing to have, right? And so just perspectives. So, and as Moira said, we just got a USDA NIFA grant approved in the beginning of this year. And now the next step is actually to try to bridge this gap we have between this complex genetics of polyploids in practical situations. That's, I'm going to do everything that I can do to try to bridge this gap. I know this is very difficult. And the way I'm trying to do this is to try to build user-friendly tools, not just to have these fancy algorithms and you can't have a user-friendly tool, right? And my intent, what I want to do with this is help breeders make this informed decisions, you know, based on their data sets. And I know I'm not a breeder, but one of the things that I'm doing right now is to communicate with a lot of breeders. I'm going to the field here with Craig Yancho, and at least once or, no, not once, or every 2 weeks, I'm going to the field and discuss with breeders what they need and what would be the best way to do this. And you know, we want to provide, based on this architecture, we have a tool to help breeders to select individuals, to mating strategies or breeding strategies using simulations. Of course, if you have the model, you can do forward simulations. And this is very important because this is very crop specific. As you saw, sweet potato is an autopolyploid. And I know that, for instance, brachiaria is not an autopolyploid, it's a segmental polyploid. So maybe this strategy is different. So this is so crop specific. Some plants are more resilient, some plants are more recalcitrant. It's very difficult to have a single way to deal with those plants. And I would say that I can have my overarching objective is to use this technology and use this inheritance in this inference of this genome inheritance or this haplotype transmission through this pedigrees to elucidate the genetic relationships between these complex individuals, using this in a QTL scenario. And with that, I'm open to questions. Here there's some collaborators, and I have so many collaborators, and I'm very grateful to work with these guys. And here I have my former supervisor, Augusto, the postdoc that is helping me a lot, Gabriel. Here we have Craig, and here we have Zobang, and here we have Guilherme. And then this is a nice picture you took in Africa several years ago. And that's it. Thank you. for listening my presentation, and I'm open to questions.
Thank you, Marcelo.
I hope yeah.
Okay, are there any questions from the audience first?
Some references.
I'll also look at chat.
Yeah, oh, the chat, of course. Okay.
Ed made a comment, so cool to see the multivalence, beautiful.
Yeah, it's nice. I love it. Yeah.
Marcelo, I was kind of wondering how important when you do some of that initial ordering and trying to create linkage maps, how important is the physical genome, or can it be the physical genome, and how important is the ordering? Is it, if it's 10% wrong, how big a problem is that? Or computation. I mean, I know you could drive everything totally without a physical genome, but I'm sure it's got to save computational resources if you have some hypothesis to start out with.
Exactly. That's a very, very good point. And since this was like a pilot project, what we did both ways, right? And we don't have the actual hexaploid genome. We are working in the hexaploid genome now. So these are reference genomes, they are diploids. And what we did was to build the de novo order. And what we observed is that in terms of phasing, it's not that a problem. Because when you switch a phase, we are switching the recombination counts. And when you have a wrong phase, your map inflates crazily. It's like you have a huge inflation, so you can see that's not, the phase is not correct. Right. So this is one thing. Of course, the local ordering is not that good. I have the graphics, and I can show you in our conversation this afternoon. And then, but they are very consistent with the genome, But you can see huge rearrangements between the diploid and the hexaploid genomes. And what I did was to manually cut these pieces, right? Compute the likelihood of our de novo order and the likelihood of the genome-based order. And all the time, the likelihood of the genome was much better. And the map was much smaller. Okay. So answering your question is very important.
It does help reduce the problem space.
Yes. And if you combine both, you can correct your genotypes much better.
But the fact that the beauty of you being able to phase there's is lovely to see that.
Yes, it is.
I've also noticed that, at least when we've talked to our breeders about the alfalfa and the blueberry, the genetic map versus the physical map, the appearance of the centromeres are extremely clear because they don't change in genetic distance, but they do in physical distance. So that's been very helpful for us to decide where markers don't need to be and where we can redistribute markers. So
yeah, we did the same.
But you need to have a good reference genome. I think that's very helpful.
Yeah, exactly. And the problem with the sweet potato is that the centromere is all over the place. It's not like a metacentromeric species. And then sometimes you have like an acrotelomere, and then you have in the closure the telomere, the centromere. So, but still, we can observe the same in sweet potato. This is the beauty of the map versus the genome. And also, there's a function in map poly that draws that comparison.
Yeah, that's been very helpful to communicate to our breeders. Any questions from the audience here? Okay, well, thank you so much, Marcelo, for your talk today. I'm sorry we couldn't have you here in person. Would have been very nice, but I really enjoyed your talk, and I'm glad you started with the most difficult species, sweet potato, because it makes everything you do in blueberry and alfalfa look super easy.
Yeah.
All right. Well, thank you so much.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.